Hey guys, welcome to the last topic in chapter 17. In this topic, we're going to bring together some of the ideas we've been talking about and talk about how muscles contract. And we're going to look at this from a cellular level, not necessarily the level you've looked at from A&P where you look at the entire tissue. We're going to look at very small areas of muscle tissue and muscle contraction. So here's our topic outline. If you have any questions about any of the stuff that we go through here, please let me know. But we're going to just kind of review muscle cells, talk about the filaments involved. And while I said that this is a review or kind of the bringing together of the previous three, we're only going to talk about actin as the filament is the only, or actin filaments are the only filament that we've previously talked about that we'll be talking about in this topic. But that doesn't mean that the ideas that are involved here aren't similar. So it's really important to understand that. Here are the topic objectives. I expect these ones to be maybe the most difficult objectives of the chapter for you, so please let me know if you have any questions about them so we can go through these. Now, as I said, in A&P, you probably went through all sorts of different stuff, but there's three main types of tissue we're going to talk about here for us because we really, once again, we're in cell biology, we're looking at the cellular level. So we're going to just talk about the three main types of muscle cells: skeletal, cardiac, and smooth. Skeletal muscle is what allows for voluntary movement. This is what allows you to tap your foot. So it allows you to take notes. Cardiac muscle, hopefully fairly clear, allows for your heart to work and pump blood throughout your body. And then smooth muscle is responsible for the involuntary movements. This allows for you to move food through your intestines, things like that. They all rely on a similar function to contract, but there's going to be some differences, but we're going to focus on how skeletal muscle works here. And at the end, we're going to talk a little bit about the differences, but the big thing I want you guys to take away is how the skeletal muscle contractions work and not worry too much about how the smooth muscle works differently. All right, so let's get started with skeletal muscle cells. Now, every time we talk about a cell in this class so far, it's been the single cell, single nucleus, and everything like that. Well, skeletal muscle cells break the rules. They merge and fuse a whole bunch of small cells together into the one kind of supercell, essentially. And it has a whole lot of nuclei in it. It becomes what we call multinucleated. And they make these tube-like structures that you can see here. Now, what they have within them when they're cytosol are these myofibrils, and they can actually run the entire length of the cell. Now, within these myofibrils are these chains of sarcomeres. And we're going to talk about the structure of these sarcomeres here in just a second, but that's what gives it that striated look that we're so familiar with in skeletal muscles. And you can see this example here with the sarcomeres. And so it's a repeating pattern that will run all the way down these myofibrils, which run all the way down the skeletal muscle cells. And that creates this tube-like striated look that you can see here on the cell. So the sarcomere is composed of actin and myosin. And so put your sarcomere thought on hold for just a second. Let's look at these components and we'll go back to the main structure of the sarcomere. So we start with these myosin filaments. You can see that in green here on this slide. And they have these two globular heads on the end, and then their tails come together to form a coiled coil tail. And keep this in. And they sit in between these actin chains. Now these actin chains, remember, they have polarity, and each actin chain is going in an opposite direction on either side of this myosin filament. And these myosin heads will attach those actins. And what this allows to do is it allows for this contractile force to be generated, which allows those actin molecules to slide past each other without actually coming into contact because the myosin filament is in between them. And we're going to talk about how these myosin heads work in just a second, but it's really important to understand that. So going back to the sarcomere, you can see this here where you have these thick myosin filaments. Now, we talked about just that dimer there just a second ago. In this case, we have a whole lot of them. There's almost 300 heads that attach to various places on these actin molecules. And this is what allows for the rapid movement. And this is a sarcomere here. You can see the Z discs with the actin attached to them on either side, and then the myosin filaments on the end. And what happens is these myosin heads are going to help pull the actin in closer together to form a contraction. And then when they release it, it allows for the relaxation. And when all of these sarcomeres work together, creates a muscle contraction across the entire tissue. So let's look at the action of the muscle contraction on a little bit more of a molecular basis here, or deeper level here. And there are 5 main steps, and so we're going to go through each of these steps on the next couple slides. So the first thing is that we start off with the myosin head attached to the actin molecule. It's pretty stiff. This is known as the rigor conformation because it's with the rigor mortis and how stiff everything is after death. It's an actively contracting muscle. It's a very short-lived state because it's going to be moving pretty quickly, but if it's not actively contracting this muscle, it will just stay in this position. So what we have happen first is ATP is added, and what it has is it's got this section on the back of the myosin head that this ATP will bind to. This creates a slight conformational change that releases that myosin head from the actin molecule. So what then happens is this ATP is hydrolyzed by the myosin, but the ADP doesn't come off. It's still there, but it's broken down. But what this creates is this cocking motion. So the myosin head's now going to move about 5 nanometers in one direction. So now it's completely shifted in its little shape. And the ADP and the inorganic phosphate's still there. As the inorganic phosphate comes off, this allows for this weak binding, because remember is that even though it came off the actin before, doesn't mean that there's no attraction. There's still this weak attraction that you can see. And so what happens is this weak binding the myosin head now attaches to a new actin filament, about 5 nanometers away, because that's how far the movement of this head was. And so now it is attached to this, and the ADP is going to come off. And what this is going to do is it's going to cause the power stroke. So what happens is as that ADP comes off, the myosin head springs back to its original position, but it takes the actin along with it. And this allows for that actin to move. And remember, it's about 5 nanometers. Now, when we consider there's 300 heads, this allows for a whole lot of movement at once and very quickly. And this happens over and over and over again. And remember, this is happening in several heads on each level, and then there's a bunch of these within each sarcomere, and then there's a bunch of sarcomeres within each muscle cell. In each muscle cell, there's lots of muscle cells in each tissue. So imagine just the massive amount of scale that this event is happening on. So it's not quite that simple, because as we know in cell, everything starts with signaling. And so what starts this molecular interaction? Well, in this case, it's the role of calcium in contractions. What we have is a signal that will come from the nerve cells that stimulate this myosin action. And so we know how the signal travels down the nerve cell from what we talked about before. And it triggers an action potential change in these special structures known as T-tubules. They're almost like veins that run through through the muscle cells. And these electrical signals are going to reach the sarcoplasmic reticulum. Now, the sarcoplasmic reticulum is this area on in the endoplasmic reticulum that's specific for muscle cells that's high in calcium. And what's going to happen is when the voltage-gated calcium channel in the T-tubule opens, it will send a signal across to the sarcoplasmic reticulum, which signals calcium release. And so now calcium is going to be released into the cell. Calcium will then serve as a secondary messenger, similar to the CAMP that we talked about in the intracellular signaling chapter in chapter 16. So what happens is calcium. Well, this calcium is going to travel to two molecular, they're molecular switches, and there's two parts to it. There's the troponin complex and the tropomyosin, and you can see this here on this slide. The tropomyosin have these long regions that cover up the actin. This prevents the myosin from binding to it. So when this tropomyosin is in this place, the myosin head can't bind and can't cause contractions. When the calcium binds to the troponin, these tropomyosin regions will move, allowing for the myosin heads to attach and create a contraction motion within the actin. And so this is where calcium is, and as calcium comes back off, they move back, as prohibiting the contraction from continuing. So it's important to see how this works. Now, if I went through that too fast, let me know and we'll go over it a little bit more in class, or feel free to rewind again. But make sure you understand this. We talked about this in chapter, I think it was 15, earlier chapter. We talked about the importance of calcium. This is once again showing us just how important calcium is to our bodies. Now, as I said, it's important to remember that each of our different muscle cells have specialized muscle contractions, especially smooth muscle cells and non-muscular cells. They also contract because they have these functions, but they use a phosphorylation process. I don't want you to worry about that here. I just want you to focus on skeletal contractions, because that's really the big highlight of what we're talking about with this chapter. So, and it's just really important to understand that the cytoskeleton can have highly specialized function depending on the cell type. And that's really a big takeaway from this. So that's the end of this topic. It's the end of chapter 17. Hopefully everything is relatively clear, but if you have any questions, please let me know and we'll go over them some more in class.